नहीं नहीं मेरा घंटे का ही कम है मेरा नहीं नहीं Hello class, very good evening. There was one emergency came to my house, so the internet was not working, so I just came to my neighbor's place. I hope it works here. So today is the last day of our class and hope it works as it should be, like the normal days. Suddenly when I start to stream, internet was like gone for without any reason. So now I'm at the neighbor's place and our voice is okay. Do I have to be loud or everything is good, right? So I have to set up everything here. No, it's clear, sir. So, okay. So, shall we start or we wait for some more students to come? Let's wait for a few minutes. I hope you will all send me your assignments and the projects that I have assigned you. So some of students they are sending daily to me, some are not. So please, at the end of a workshop, do send me all your things that are required. Uh, So we start now from bioinformatics class. Uh, So welcome everyone. So today is the last lecture of ours as we are done with the syllabus already with the all course content. Like I found yesterday our course content. It was lying somewhere in the our byte, I guess. Or hmm. So this was I think was for the plant biotechnology and that I have to taught you. I hope we have covered everything. Each session has to be a 2 to 3 hours, but unfortunately I didn't thought that the lecture will be so short because when we do them at the university level, so each lecture used to be at 1 hour and we don't know how much time do we take, but being online things goes really fast because of this virtual world. uh, so that might be the reason. So my calculations goes up and down. Sorry for that. And but anyhow, our task was to get to know about all these topics. I will also share these topics with you so that you know what you did uh, from this workshop, and you can remember for the future work also that during this workshop all these aspects were covered very crisp and uh, yeah, that's great, Gaurav. Thank you so much. So how many days we will get more to complete our review? It is on the process. I mean, it should be 5 days are enough actually. If you start for something, if you put every day few hours to it, so 5 days will be enough to go for it. So that will be not much work. And publish them also. Some of my students they published in free journals, even though they don't have much values, but it's good learning that how you could publish your own journal and get the benefit out of it. So that's the beauty of your uh, being connected to journals. So bioinformatics, this is a chart that you can see which has major things coming out. It's mainly dealing with protein structure, algorithms, computational data, process, analysis, complex prediction, homology, genome, DNA, one genome, various things. So all these things are covered in this. Uh, So we will cover in this topic some biological concepts that we already know much about DNA and RNA, so we will not go through it, but a brief history of a young field of bioinformatics, where this all data is coming from, and large scale biology, new words, new techniques that we will be dealing in this topic. So what is bioinformatics? It says in the Venn diagram, you can see very nicely that is it's a mixture of biophysics, mathematics, statistics, informatics, biology. So when we bring them all together, biophysics and mathematics and biology, and then study of all these things together is known as bioinformatics. Further, we can see uh, that there is a we know this central dogma from DNA to RNA, from RNA to protein, then it goes to check the phenotype or proteome. So beyond that, there is also thing called DNA genome databases coming from our DNA part. So that's the major thing that is bioinformatics is consisting of to analyze these genome databases with next generation sequencing like Illumina, Sanger technique, है ना? So all these techniques are quite important in day-to-day life of our next generation sequencing. Further from RNA, you can get the complementary DNA transcriptome data you can get, and from the protein you can get protein sequences, structural databases. All these things could be attached to it. National Center for Biotechnology Information and Definition. According to the NCBI, the definition of bioinformatics is it's the field of science in which biology, computer science, and information technology they are all merged together to make a single discipline. So the ultimate goal is to field to enable the discovery of new biological insights as well as to create global perspective from which unifying principle in biology could be described. So most of the time, whenever somebody is in the masters, like in one of my friend was telling, when she went for studying in the Lund University in Sweden for her masters in bioinformatics, most of the students were from the informatics in bachelors, and no one didn't know anything about biology. But somehow they get to know about biology in later paths. So that's how information technology, computer science, and biology we merge them all together. That is where is bioinformatics is there. So there are three important subdisciplines within bioinformatics. That is development of new algorithms and statistics, analysis of interpretation of various data types such as amino acids, protein domains, protein structures. Furthermore, the development and implementation of tools that enable efficient access and management of different types of information. So all these three parts they make all together your bioinformatics. Furthermore, there in bioinformatics we also take care of the phylogenetics overview. That is tree of life. Tree of There are mainly three domains of life are there in the case of bioinformatics. So that is um, archaea, eukarya, and bacteria. So these are the three major regions that are concerning here with us. And furthermore, within the archaea, there is Canaro architeo. In the eukarya, you can see there are many from cholanoflagellates, red algae plants, all these parts belong to the eukarya. And in the bacteria, they are Thermotige, VS6, Firmicutes, Chlorobi, TM7, cyanobacteria, chloroplast, all these includes in bacterial part. This is a traditional way of showing it of microbial diversity, tree of life, come of age. There are two types of cells, as we know, bacterial and eukaryotic cells, and for the both of their genomes are quite different. Their working of things are quite different, and their nucleic acids, the nucleins are quite different. So based on that, there is a central dogma is also different from the molecular biology point of view of prokaryotes and eukaryotes. So here we can see in prokaryotes you have gene A and gene B, which give rise to transcription, that is coding region A, coding region B, and these coding regions will produce mRNA, and this mRNA with different coding regions they produce protein A and B. Further, uh, in the eukaryotes, they are exon in the DNA. They are exons, intron, exons, intron, exons. And when the transcription happen, first primary RNA transcript is produced with the exon, intron, and exon, intron. But after the RNA processing, introns are removed. They are exercised during this process. We remove these introns, and mainly mature RNA is left in the nucleus. And this is then transported to the cytoplasm. You have a mRNA, and then you could go for translation for protein and cytoplasm. And this is shown you to the alternative way of splice exon, whereas everything is separated by 300 naught, and hence at the end, because of splicing code, you have predicted change in the exon inclusions. So in the protein splicing, there are intervening sequences also are present, whereas in RNA it's exon, intron, and exon. Where mRNA splicing happen, you have a mature RNA. Same way, there in the protein we have extine, intine, extine, and inteins are removed, and then hence at the end you have mainly mature protein afterwards. The same way like mature mRNA. So bioinformatics is mainly about genetically coded information between your DNA and RNA and proteins. So bioinformatics deal here about your codons, anticodons, your DNA sequence data. That is, if you can see that there's a DNA sequence with TTT, GTT, AAT, CAG, CAT, CTT. Then after transcription, there is a thing called, we have mainly the anticodons for that. That is UUU. From the anticodon, with the help of transfer RNA and ribosomal RNA, your amino acids are produced. Phenylalanine, valine, aspartic, this I don't know, sorry, glycine, histidine, leucine, then COH, then on the other side is your amino group, and hence your protein is produced. So mainly the bioinformatics is dealing with these sequences, with the how many sequences are there, how many things are working out. So it is about data management, databases, database structure, analysis of interpretation of data using web-based or stand-alone tools, writing codes like Perl, Python, Java. So mainly now Python is major language that is being used in biology to make programs, to solve your daily problems, to make algorithms, to make statistics, to make machine learning also is also being played in the biology. So they make some programs which will solve your all daily problems on a daily basis. Whether it's a calculations, whether it is anything that is to deal with big data. So it will be all done automatically with the creating one simple programs which take some time, and you can have your results from it. So in summary, bioinformatics help us to uncover biological information hidden in the mass of sequence, structure, literature, uh, and other biological data, and obtain an insight into the fundamental biology of cells and organisms. So like from genome we have transcriptome, from transcriptome we have proteome, then metabolome, then phenotypic structure out of it. So as the life get complicated, the data structure analyzing is also complicated. So this is the computational tools and infrastructure are being shown here. On the y-axis, there's a biological complexity. So initially, comparative genomics is quite easy to go for, but as we go up with muscular machine classification, stimulation, protein machine interactions, community metabolic regulatory, the complexity is getting increased. And then molecular based cell stimulations. With the help of various softwares, you can do it. So it's getting, yeah, it's getting complicated as we go further. Further, the data DNA, so raw sequence information, there are four bases, whether they are coding or not, to check them, whether these are parsed into genes when or there will be any signals or patterns. So all these things matters in collecting the DNA data when you're collecting the data for DNA. So restriction enzymes are as we know, they are the sequences where they cut specifically. So E. coli, for example, it cuts at a six base pair sequence GATTC, and how often would expect to find recognition sites? How often it will recognize these sites? So in this case, GATTC in this amino acid sequence, um, your bioinformatics will play the role in that case. When it will cut, so you need to have full sequence of amino acids, and to cut it from where, and all these things are being played role in by them. So I will not repeat. So this is same old thing that we did. Purine bases, they are adenine, guanine, and then pyrimidine bases are thymine and cytosine. Base pairs within the DNA double helix. A forms two hydrogen bonds with T on the opposite strand, and G forms three hydrogen bonds with C on the opposite strand. This is the genetic code, U. So this is how you make them. You write UCAG on the first letter, then second letter UCAG, then the third letter UCAG, UCAG, UCAG. Then you take first letter from here, second letter from here, third letter from here, and then you have 64 triplets like this. Whereas your methionine is the initiator code, and your stop codons are UAA, UAG, UGA. So all these are your stop codons then. So rest are your genetic codes. So these are all also important while analyzing the data. So these are the protein sequences, taxonomic distribution of sequences in UniProt or Swiss-Prot. This is a universal data, UniProt data. This has been shown in being issued in July 2017. Contains around 5, 55, like how much it is? 5,55,100 sequence entries, or 5,550 100 sequence entries, comprising around these much of amino acids abstracted from those references. So you can see that um, the bacteria is the largest with 60 percentage. Then comes the archaea, then comes your viruses, uh, then comes your eukaryote. I think archaea is 4% and eukaryote is 33%, because this is not possible in that case. Yeah. So this is then data, DNA, RNA, protein structures could be predicted. This is the PDB. This is protein database. There is a website called PDB where you can find the molecules, the structures, how the structures looks like. So these are various aspartate tRNA. This is phenyl tRNA with the anticodons at the bottom, as in AAG, CUG, and these are the basics of your. Uh, uh, Yeah, so bioinformatics also plays role here to identify the structure of your proteins. So that was some introduction so far. Now let's go for a little bit about historical landmarks. That is, it started in the 1953 with the structure of DNA revealed. Then the first protein peptide sequence was identified. Then 1960s, first enzyme sequence, elucidation of the genetic code. Then DNA sequencing, first human GNM was isolated. Then gene bank database set up. PCR was described in 83. Then fluorescence-based DNA sequence was done in 86. Then first draft of human genome was done in 2001. In 2003, the Human Genome Project was completed. So like from 1953, structure of DNA was discovered, and by 2003, Human Genome Project was completed afterwards. And bioinformatics includes your computational biology, computational molecular biology, biomolecular informatics, biocomputing. All these things are included in this. So their results by year, we can see it here. That is every year. So it's increasing like this. By 2003, as we know that in 2003 this Human Genome Project was identified, but since 2003 this field is increasing massively, huge. It started by 2010, it's just got the peak, and it's keep on increasing then later on. That's how the PubMed research, if you put bioinformatics, a lot of studies are being done after the 2000 years. So historical landmarks afterwards also a little bit more. Needleman and Wunsch, they did the algorithm for sequence comparisons. Further, Chow and Fasman predict the protein secondary structures from the amino acid sequence. They could predict the secondary structures just because of the amino acid sequence with the help of prediction softwares. Smith Waterman, they identified algorithm for sequence alignment. There was some hydropathy plots were created. Further, FASTA, similarity search of sequencing database to check the whether two sequences are similar. FASTA was created. Then basic BLAST was done in 1990s to check the rapid sequence comparisons between two different species. BLAST is done for the same. So there are now more than 100 million DNA sequences stored in the gene bank database. So we can see that uh, in the 1982s, so there were like 606 sequences. In 1990, they become 39,000, and by 17, the base pairs and sequences, they abruptly increases. So this is a growth of gene bank has been shown to you from 1998. Like it started to abrupt up, 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 up. So from journey of 1971 till 2013. So how things are working on in the case of gene banks for us. Then further, National Library of Medicine, 24 years of growth and CBI data and user services. What they have identified here is uh, pub central pub these are the various gene bank pairs in the blue. So these are gene bank base pairs, and the red bar is the users who are using them, average users. So these are the various years when the various identifications were found. So BLAST was found in 1990, then Entris was in 1991, Gene Bank in 1992, Genomes in 1995, then OMIM, PubMed, Human Genome, PubMed Central, PubChem, NIH, Genome-wide Association Studies. Um, so all these things, MedGen, PubReader, so they were created in the following years, and how gene bank base pairs are increasing with the users of the same also. So users of these various softwares also, these channels, these platforms, their users are also increasing with the years. So these are our all NCBI users weekday per meeting. So as you bring the new things, people also come and check the things like that. Then comes your DNA sequencing with the help of Sanger, who have created a technique with the chain terminating inhibitors. So it's a high throughput technology, such next generation sequencing we call them. So first sequence was bacteriophage, then the first living organism Haemophilus, and then first eukaryotic sequence was Saccharomyces cerevisiae, Baker yeast later on. Sequence capabilities versus computational power. So these were the sequences done with the blue bar shows the sequences done with the techniques, but these are computational technology. Then they with the computation in the years they were also doing it. So somehow they were going in parallel. Initially, you can see that bases per day with the computing were more, but as the next generation sequences got rise in 2006, so it just pick up the pace, whereas computing got bit low, not so much high. So next generation sequencing somehow play overrule the computational power of bioinformaticians, but still it was higher in the initial years, but just now it got decreased. Further, sequence read archive database growth is showing you. So the sequence read archive SRA stores raw sequences data from the next generation sequencing technologies. So they were also increasing in exponential curve like this from your year 2010 or before that, then going by 2015 going up and up and up. Then comes the complete genome sequence available in the public domain, which has been more than 10,000 complete genomes published. And this graph showing us of the bacteria, eukarya, archaea from the 1974 when the Fred Sanger method of sequences was found out. Then the various species from C. elegans, S. cerevisiae yeast, D. melangster, Homo sapiens was finished 2003. All there are completed like that. So everywhere in each part, the bioinformatics was playing big role in our research of computational biology of genomics, or is it proteomics or transcriptomes, anything. So bioinformatics has been always been there to help the scientist, which might be not possible while working in the wet lab. So Human Genome Sequencing Projects, the Human Genome Project announces completion of DNA sequence of Human sapiens. So, so chromosomes like chromosome 21, 22 were discovered in 2000 Nature paper 2000 and in 1999. And this is how the first Human Genome Projects, the booklet looks like when it was done. It's such a big series of uh, yeah, books were there. So one could get to know about the human genome with which sequences at which level. So like 22 or so, there are more than that. So on some new methods, new words, the last topic of our bioinformatics chapter. So genomics refers to the analysis of complete DNA sequences. The study of obtaining functional genomics includes study of obtaining overall picture of genome functions, including expression profile at the mRNA level and the protein level. And whereas proteomics refers to a study of proteomes using technology of large scale protein separations and characterizations. Whereas transcriptome is the complement of mRNA transcribed from the cell genome, whereas transcriptomics is the generation study of complete mRNA expression profiles. And further, structural genomics or proteomics is assignment of three-dimensional structures of proteins products to investigate their implication of these assignments and provide probable functions and evolutionary relationships. So hypothetical proteins, paradoxins, all are being discovered uh, cause of proteomics. So omics expands. So and genomics is like quantitative study of genes, regulatory, and transcriptomics is the RNA gene expression. Proteomics is the protein expression, whereas metabolomics is the metabolites and metabolic networks. Pharmacogenomics is the quantitative study of how genetics affects responses to drugs, and physiomics is the physiological dynamics and functions of whole organism. So we have discussed the transcriptomics already. So this is one example showing us from the cell sample you took the cellular RNA. Then either you can do the micro approach or RNA-seq approach. In micro approach, you label them with the complementary DNA or RNA and then infer their transcript to analog the quantification. That how much is it green label or red label? So DNA micro approach. That's a whole new chapter, that's a whole new technique. Sometimes we discuss this later part. And then comes the RNA sequence approach. This is we do the complementary RNA DNA, sorry, and then compare with the target genome and enumerate the sequence out of it to check the digital quantification. So in the microarray approach, so once we have a bacteria growing in culture and we label with the red dye, red Cy3 dye, and a bacteria from the site of infection, where the infection happened in the human body, and we label it with the sapphire dye, green sapphire dye. Then we co-hybridize these two samples and check at the DNA microarray uh, sheet. So RNA binds to corresponding genes. So blanks are the ones which are not expressed, greens are the ones expressed in the infections only. So green genes are the one which are expression only, red expressed in the culture only, and yellow expressed in both culture and infection. So we can see that how many which gene. So these dots are corresponding to one gene, and we can say that this gene is expressing in the culture only, and that yellow gene is expressing in both. So we can say that with these yellow marks that these genes are being present in the infection of the human body, and also in the growing outside in the culture, that infection that it was producing. So both could be the gene that was responsible for the uh, disease actually. So we can calculate like this. Then RNA sequence is a isolate of all mRNA, convert them into complementary DNA using reverse transcriptase and sequencing the complementary DNA, then map sequence to genome. So in this case, we have our mRNA. mRNA is fragmented by 50 to 300 base pairs. And then reverse transcription is done. Then sequencing is done to check the full spectra of a protein. And then fragmentation is done. Then MS further MS data is done of your data to check the peaks, like how the peaks are coming out. Then you can quantify this full spectra, or you can identify further the genes what you're looking for. So they're divided into three parts of your proteomics. First is sample preparation, then the mass spectrometer, and then the data analysis. So from one gene sequence to one predicted amino acid sequence to many proteins. So from gene, we predicted translated gene products and co and post translation modification of the proteins. From N or C termination, glycosylation, and other modifications are being done then further Also, it helps in the system biology, like this organism. First, there is an planet, then from planet is ecosystem, from ecosystem there is a community, then community is organism, then the cell, and then DNA. So, how the interrelationship between biological complexity to identify that is known as protein interaction system biology. So that's a user. That's something about user interfaces between data hubs and methods. How it is done with the QTL maps. So there are top 10 challenges for bioinformatics. That is a precise model where the when transcription will occur in a genome, and precise predictive models of alternative RNA splicing, precise model of signal transduction pathways, determining the DNA protein recognition codes. These are the top challenges when you are doing so. Further, rational design of small molecular inhibitors of a protein, mechanistic understanding of protein evolution, mechanistic understanding of speciation. Further education development of bioinformatics curriculum is also required. So that's it. That's the end of our lecture for today from the bioinformatics perspective. So here you can have a 5 minutes break. So we meet us at 10:40 or so, or 10:45, 10:40. Let's meet at 10:40 then. Sorry, any questions you have, students? If you have any questions, please feel free ask. So it was something to tell you about how bioinformatics has a key role, playing role in many aspects of it, and how it could be done, and how important it is. So, so okay, I take break now. I just come by then. Yes, students. Tell us what. बच्चे blast fasta uh, is just to check between two amino acid sequence. Uh, so you can also do fasta. So if you wanna check blasta sequence nucleotide. So you can have two different sequences, like one from monkey, one from human, and wanna check how much sequences is similar. So you check the nuclear sequence by doing blast. So I don't have much example. You can also do FASTA the same way by adding the accession number or the FASTA of both species, and then you can check it in this case. Um, so on that note, so what was the other question? By which way you have to send the assignment? So assignment date बच्चों is your today is 14th. So I can give you 5 days more. 5 days will be a lot actually. So I have given you plenty of time. So maybe okay, by 18th of September. Send your review. This is for the review. 18th of September. Assignments you can send anytime. Because this was not compulsory, you will get your certificates even though you don't send me assignments. But I love to see technique also. So this is including everything, that is protein isolation, quantification and purification. So this technique includes, so we will deal with this technique is assay, homogenization, fractionation, centrifugation, quantitation, chromatography, and electrophoresis. So I have divided this chapter with lot of videos also. So and this will be a lot of interactive sessions. You will have a lot of animations also and a lot of videos also. So this will be quite an interactive session for you today. So some names about the Nobel Prize winners who did work on the chromatography and electrophoresis, that is Archer John Porter Martin. He worked in 1952 on the field of partition chromatography. Then that Richard Lauren Millington Singh, he worked also on the invention of the partition photo chromatography further on. Then comes your Arne Willem Corinthius. He worked in the Uppsala, Sweden. His work was on the electrophoresis and absorption analysis, especially for his discoveries concerning the complex nature of serum proteins. Then comes your fan. So now these are the scientists who are famous for the mass spectroscopy and sedimentation technique. These are various Nobel Prize winners. The first one who identified the biological molecules, John B. Fan. Then Koichi Tanaka from Japan. He discovered the soft disruption ionization method for mass spectrometric analysis of biological micromolecules. Then Theodor Swedberg, he identified dispersed system. Based on his Swedberg unit has been created in 1926. Then comes the Nobel Prize winners who did the organelle fractionation. So Albert Claude from Belgium for discovery on the structural function of the cells. Then Christiane de Novo, Rockefeller University, and also George Pallade. So they all three got for the discovery of organelle fractionation. Then your Frederick Sanger for his work on the structure of proteins, especially that of insulin, and this is another Frederick Sanger who contributed in the determination of the base sequences in the nucleic acids. So, something about protein assays. So this was about the Nobel Prize winner, somebody who really, so this field of proteomics and protein isolation and purification has been always been in charm. And one of the difficult technique though, but once the results are there, then it's a eureka. You are into the new world. So assays, they are the biochemical protein assays used to monitor the presence of a target protein, such as enzyme activity, specific label, functional properties such as binding. All are done with the help of this technique. So there are like lactate is converted into pyruvate with the help of lactate dehydrogenase. So you can check it by using this enzyme by changing the color or by changing the concentration or spectroscopy in different ways. So also ELF 5A precursor is converted to the deoxycyl ELF 5A in the presence of deoxypurine synthase, and further this could be presence of deoxy hypozine hydroxylase. It could also the various techniques to check it. So further, uh, just a second. So further, the right hand side you can see there's a homogeneous form of your mixture. We centrifuge at 500 G for 10 minutes. So you have a supernatant and pellets. In the pellets, you can see your nuclear fractionation. Your nuclear part will be there. Then you again do 10,000 G for 20 minutes. Then you have again in the pellet is the mitochondrial fractionation. Then further at the 10,000 G for 1 hour, there is a cytosol soluble proteins, and at the pellet is your pellet microsomal fractionation. So at the end, so you can get it by keep fractionating, by keep centrifuging it, you keep getting a new different part of your cells. Further, to isolate your protein, centrifugation technique also helps to separate particles and macromolecules based on the mass, density, shape. So here differential centrifugation has been shown to you that more dense the particle, less dense particles are present. So with the help of centrifugal force, particles settles according to their mass. So the most dense particles will be down, where the supernatant will have the less dense particles, and you decant your liquid into the container and you can have it like this, shown it to you here. Then rate zonal centrifugation, sample is layered on top of gradient, large particles, smaller particles. So sucrose gradient is there and this is the upper part. Then centrifugal force is done, particles settle according to the mass. Then stop centrifuge afterwards. Then you can separate your solutions by collecting at different parts of a solution. So these are the various fractionation part of, according to their density. They are changed, and you can and this is the increased mass of particles. So first is nothing, then there is something, then there is something, then there is something. You can collect and then to do two different assay. So protein isolation centrifugation, there is like 3000 G for 5 minutes. So large organisms will be present at 10,000 G for 10 minutes. Mitochondrial inclusion bodies will be done for 4000 G for 30 minutes. Membrane, plasma membrane will be done. Then 200,000 uh, large protein complexes at 400 G for 24 hours. Protein of the size of 50,000 Dalton will come. So sedimentation coefficient looks like m mass 1 minus mu square by psi divided by the frequency mu, which is equivalent. And you have to just remember in this sedimentation coefficient is that one sediment coefficient is equal to the 10 raised to power minus 13 seconds of total time. So according to this formula, sedimentation velocity technique, you can separate your molecules according to their sediment coefficient, and you can have different particles as per the things. So here you have a centrifuge tube, and this is your low density solution. This is high density solution, and then you have your centrifuge tube here with a density gradient in it, and then you layer your samples on top. Then it's go for the rotation, and separation is done according to the sedimentation coefficient. So you collect each part according to its weight into different tubes like this. So these are told you that like various sedimentation coefficient, you can have the density of your molecules according to different density. You have your various sedimentation coefficients are happening. So microsomes, most viruses, chloroplast, nuclei, ribosomes, DNA, RNA, all these are the sedimentation velocity techniques and done as per the things shown to you. Then comes the first part of your quantification, like you want to quantitate how much protein is being produced. So we have isolated our protein with the help of various techniques, various part of the technique with the help of Bradford protein determination. The technique is quite simple and faster than the Lowry method and subject to have less interference. In this we use commonly blue dye, which appears to be most readily to present the agenyl lysyl residues. This specificity can lead to the variation in the presence of the assay to different proteins. So let's have a video about that now. It's video time. So I will not speak. I think if I have to speak, I will let you know when the video is playing.
In this video, you will learn protein quantitation using the Bradford assay. Protein quantitation is one of the most commonly performed procedures in a biotechnology laboratory. In this activity, the protein concentration in milk will be measured using the Bradford assay. For this activity, you will need 1x Bradford reagent and 1x phosphate buffered saline, or PBS. Using a 20 to 200 microliter micropipette, set the pipette to 98 microliters. Pipette 98 microliters of 1x PBS into an empty microcentrifuge tube labeled sample. Switch to a 2 to 20 microliter micropipette and set the pipette to 2 microliters. Pipette 2 microliters of milk sample into the microtube labeled sample A. Mix the milk sample and PBS by either pipetting or vortexing. This mixture has diluted the milk to 1 part in 50. Label 2 cuvettes, one control and the other sample. Do not handle the cuvette where the light passes through and make certain your labels are well above this area. Pipette 20 microliters of diluted milk sample into the cuvette labeled sample and 20 microliters of 1x PBS into the cuvette labeled control. Next, we will set up protein standards. See your protocol for the correct concentrations. Label a cuvette for each standard and mark it with the corresponding concentration. Next, add 20 microliters of each protein standard into the corresponding cuvette. Make certain you use a clean pipette tip for each sample. Now you are ready to add the Bradford reagent to all of the cuvettes you have prepared, including the sample and control. Add 1 milliliter of 1x Bradford reagent to each of the cuvettes. Mix completely by pipetting up and down with the micropipette. Incubate the cuvettes at room temperature for 5 minutes. After 5 minutes, visually compare the cuvette containing the milk sample to the cuvettes containing the protein standards. Determine the standard that most closely matches the color of the milk sample. Estimate the protein concentration of the milk sample based upon the visual comparison. To determine the protein concentration using a spectrophotometer, select for the Bradford assay. When asked to insert blank into the spectrophotometer, insert the cuvette labeled Control. Use the blank to set the spectrophotometer to zero absorbance, or 100% transmittance. If instructed, read the absorbance of the 7 protein standards and record the absorbance values. Remove the cuvette labeled blank and insert the milk sample into the spectrophotometer. Read the absorbance and record the value.
So mainly, I'm here because it's very hard to show you these things in the virtual world, but I will be showing all you these techniques, how it is done and how it is done in the lab. But about the calculations, we will not dealing in most of the cases, but we are dealing mainly how these techniques are done manually. Yeah, we will remain till that. So I hope our connection is all good and you are all watching it. You're all on. Yeah, all is good. Please thumbs up if all is good. You are there, students. Thumbs up. Even I don't know how to thumbs up actually. I can send you smile. Okay, I continue. This is how you can quantify the DNA also by using a nano-orange protein quantification assay in which you add your dye to samples, you heat for 10 minutes, then cool to room temperature and read the fluorescence afterwards. Then you have a fluorescence versus protein in this way, your bell shape curve, not bell shape, exponential curve. Then after the quantification of a protein assay, how you can do the protein purification? So protein purification could be done by using a technique called dialysis, in which you remove the small molecules from the protein solutions to change the buffer solution of the sample solution. So dialysis tubing or bag is made of cellulose and has a different cutoff range. So here you have big molecule and small molecule in the dialysis tube, and what you do is at the start of dialysis, your small molecules will be at the equilibrium, your will be removed, and mainly your big molecules will remain inside. That's the basic principle of it. There is also Proteo plus kit technique has been done in the case of Eppendorf tubes. They have created a precipitation protocol with the help of matrix associated laser desorption spectroscopy, mass spectrometry, that is MALDI-MS, Q Biogen. So a little bit about this video now. that's it. That's how you do your dialysis of your proteins. Then comes the liquid chromatography. That is a term which refers to all chromatographic methods in which mobile phase is liquid and stationary phase may be liquid or solid. So this is further divided into gel filtration chromatography. So here you can see that protein sample with molecular exclusion gel being done, and then you can see on the right hand side that it has carbohydrate polymer beads, small molecules enter to the aqueous spaces within the beads. Yeah, the larger ones couldn't do it, so they will remain, they cannot enter to the beads. So you separate your molecules according to your beads actually. So you can see first the yellow part will started to come, then the green part will started to come, then you separate them with the flow of like you can flow with the wash buffer to remove it. So like this, in this case you can see in the gel filtration chromatography, large scale proteins and small scale proteins, a layer of sample on column, you add buffer to wash proteins through column. So you have polymer gel ahead like this, and then you can run your samples and then you collect the fractionations at the end. So this is how it is done. This is the gel they have used is Cefadex G75, in this case Cefadex G10, HB in both cases, and separation of proteins by size, gel filtration chromatography. So gel filtration chromatography could be seen here. That is brand name, polymer base, exclusion limit, Cephros, Cephacel, Cefadex, Toyopearl. So these are the various exclusion limit for the same respectively. So now let's watch how it is done, this technique.
We're starting with this is Cefadex G50. The number here gives you an approximation of the size exclusion. So this is will exclude proteins above 50 kilodaltons, 50,000 molecular weight. Inside is a white powder. In the beaker I put 9 grams of the G50 powder, and I swelled the powder overnight using this tris buffer, 30 millimolar tris buffer, pH 8.0. You can see that the 9 grams has swollen up to a volume of about 90 milliliters, almost 10 milliliters per gram of solid. Now we're going to gently stir the gel. It's important not to stir this too vigorously. Don't mechanically stir this. That can break your gel particles up into very fine dust, which will impede your flow rate in the column. So this is making a slurry. I'm using a sawed-off pipette to transfer my slurry to the column. For gel filtration, I want a fairly tall gel bed. So I'm trying to mostly fill this column. And excess buffer will drip out the bottom. So here you can see the drips coming out the bottom. Gel bed is starting to settle a little bit. Gel bed is right here. I'll add a little more buffer so that it doesn't run dry at the top. So I'm gently adding a little buffer to the top. This looks great. You can see the gel bed is fully settled here. There's a little bit of buffer on the top. I've capped the bottom right now to keep it from going dry. I'm going to put a mixture of 2 molecules onto the column. Blue dextran is a very large molecule. It's a polysaccharide. Molecular weight about 1 million. Fluorescein, on the other hand, is a small molecule. Molecular weight about 350 grams per mole. I chose these 2 molecules because they're nicely colored, so I'll be easily able to see the separation visually as they go through the gel filtration. I've drained the buffer, so now there's no residual buffer on top of the gel, and I've made a mixture of a total of 0.8 milliliters, 400 microliters of the fluorescein, 400 microliters of the blue dextran. nice green color. I'll separate these on my gel. For this size column, this is getting near the maximum I would put on the column. I'm using a Pasteur pipette to gently load my material onto the column without disturbing the gel bed. So here you can see the blue molecules, the large molecules, are moving faster through the gel. So I'll hope that I can get a complete separation between the blue and the yellow. Now the excess on the top of the gel is all soaked in. So I'm adding buffer very carefully to the top of my gel filtration column. I'll just add a fraction of a milliliter first to help get everything into the gel before I add a large volume of buffer. So now the separation is proceeded somewhat. I'm going to try to have a decently large volume this time of buffer on the top. About a milliliter would be great. If I do this well, then the buffer that I put on the top remains colorless because all the colored molecules have already gone into the gel. Alright, now you can start to see a little colorless layer on the top. That's perfect. All the colored molecules are in the G50 and they're starting to separate quite nicely. The yellow molecules are up here, the blue are down here. It's still got a little overlap. So I may have overloaded this column, but we'll see. I'm hoping that before the blue molecules elute that they'll actually be separated from the yellow, but it's going to be close. So out of the bottom now, blue liquid is dripping. You can see I do have a colorless layer starting to develop here. The fluorescein has been slowed down by the gel filtration. The blue dextran is coming through. So this technique is often used to separate a protein, which is large, from the small molecules that could be salts or could be a fluorescent tag that is unwanted. I've topped up with buffer on the top so that it never runs dry. The fluorescein is now eluting from the column. Fluorescein is fluorescent. I'm shining a little blue light, UV light on it, and you can see it glows green. The last little bits of fluorescein are emerging from the column now. Here's the fractionation. This first tube has nothing in it. So this volume, which looks to me about 2.5 milliliters, is called a void volume, where nothing comes out. But then the blue molecules, the blue dextran, came through the column swiftly. So this is about a milliliter. Some of the blue dextran tailed into the next fraction. This is low concentration. Then I have a nearly colorless fraction here, and then the fluorescein starts coming out here. Fluorescein stopped coming out right about here. So the total volume will be 2.5, 3.5, 4.5, 5, 6, 7, 8, 9, about 10 milliliters is the total volume, which is what I would expect for a column of this size. Often what you want is just this first fraction with the large molecules. If you're trying to desalt a protein or remove the unbound label from a protein, then this is the fraction that has value. These fractions would just be junk. The original mixture, you can see, glows nicely. And what I believe is isolated blue dextran from the column has no glow in the UV. That's a sign that I have successfully separated out all the fluorescein from the blue dextran. So the column, if I've done it well, comes out completely colorless. It's reusable. Sometimes microorganisms like to grow in the cephadex, so if you want to preserve your column for a long period, keep it in the fridge, certainly. And sometimes azide is added to the buffer to prevent growth of microorganisms. Here's tris buffer with 0.01% sodium azide, and this will prevent microorganisms from chewing on the deck strand that's in the column if you're storing it over a long period of time.
So that was about gel filtration. Now next comes the chromatography of ion exchange one. So in this part, mainly you have positively charged proteins bind to the negatively charged beads. So you have a beads, big beads. To that, mainly your positively charged proteins will join in, whereas the negatively charged proteins will spread it out. So that's the basic principle about ion exchange chromatography. So protein in a solution that has a low ionic strength is added to the column. The column, the protein interacts with the beads via electrostatic interactions, and the resins carrying the positive charges are called anion exchangers because an ion will exchange on this type of column. And resin carrying negative charges are called cations, and because cations will exchange on this type of column. So here is your carboxymethyl CM group ionized form, and this is your diethyl aminoethyl group, DAEA proteinated group. So in the cation exchange chromatography matrix will be negatively charged, whereas in the anion exchange chromatography matrix set will be the positively charged. So chromatography, ion exchange chromatography is yeah, it could be the group could be converted into sulfonate, quaterniumine, diethylamino, aminoethyl, or hydroxy appletate. So abbreviation is SQDEAHA, and exchange type is strong cation, strong anion, weak anion, strong anion, and these are the following structures of it. So let's watch another video
Ion
about
exchange chromatography. Process that allows the separation of ions and polar molecules based on their charge, as well as other forms of chromatography. In ion chromatography, there is a mobile phase, which generally consists of buffer solution, and there is a stationary phase which consists of a matrix that contains charged ionizable functional groups. This type of chromatography is subdivided into anion exchange chromatography and cation exchange chromatography. In anion exchange chromatography, negatively charged molecules are attracted to a positively charged solid support, which is usually beads that provide a positively charged surface, such as resin quaternary ammonium. In cation exchange chromatography, positively charged molecules are attracted to negatively charged solid support, which is commonly beads that provide negatively charged functional groups, such as resin methyl sulfate. Ion exchange chromatography is frequently used for the separation and purification of proteins. The basic process of this chromatography can be represented in 4 steps: conditioning or equilibration of the stationary phase, sample loading, washing, and elution of the retained molecules. The first step is the equilibration of the stationary phase to the desired start conditions. The pH and ionic strength of the sample buffer are selected to ensure that, when sample is loaded, proteins of interest bind to the medium, and as many impurities as possible do not bind. Therefore, the same buffer is used for the equilibration of the stationary phase. When equilibrium is reached, all stationary phase charged groups are bound with exchangeable counterions, such as chloride or sodium. When the column is equilibrated, the second step is sample application. Depending on the pH of their environment, proteins may carry a net positive charge, a net negative charge, or no charge. This net charge can be determined by comparing the pHi of each protein with the pH of the buffer. Proteins are large biomolecules, consisting of one or more long chains of amino acid residues. Amino acids are organic compounds that contain amine and carboxyl functional groups, along with a side chain specific to each amino acid. Depending on the pH of their environment, the amino group can be protonated and become positively charged. The carboxyl group can be deprotonated and become negatively charged. and the side chain of some amino acids can become positively or negatively charged due to the gain or loss of protons. So, the net charge on the molecule can be positive, neutral, or negative. The isoelectric point, or pHi, is the pH at which a protein carries neutral charge. At a pH below their isoelectric point, proteins carry a net positive charge. And at a pH above their isoelectric point, they carry a net negative charge. In this example, the pH of the buffer is equal 5, consequently, protein A has a net negative charge. And both, protein B and C, carry a net positive charge. In addition, protein C is more positively charged, since its pHi is higher than the pHi of protein B. If we apply cation exchange chromatography, protein that has a net positive charge, it will bind to the negative charge beads of the stationary phase. On the other hand, uncharged proteins, or those with a net negative charge, would not bind. They pass through the column at the same speed as the flow of buffer. When all the sample has been loaded, the next step is column washing. The column is washed with start buffer. To ensure that all non-binding proteins have passed through the column. When all the sample has been loaded and the column washed, conditions are altered in order to elute the bound proteins. Most frequently, proteins are eluted by increasing the ionic strength of the buffer, or, occasionally, by changing the pH. As ionic strength increases, the salt ions compete with the bound components for charges on the surface of the medium, and one or more of the bound species begin to elute and move down the column. The proteins with the lowest net charge at the selected pH will be the first ones eluted from the column, as ionic strength increases. Similarly, the proteins with the highest charge will be most strongly retained, and will be eluted last. Protein B has the lowest net positive charge, so it can be eluted first. Then, protein C with the highest net positive charge, it can be eluted next, by increasing the salt concentration. Also, varying the pH of the buffer can be used to effect a separation, and to elute the retained proteins. As we seen, the pHi of the protein B is equal 7, and the pHi of protein C is equal 9. So if we add a buffer with pH above the isoelectric point of protein B, and below the isoelectric point of protein C, the protein B will become negatively charged, and can then be eluted. The protein C is still charged positively, consequently, still retained. Next, protein C can be eluted if we apply a buffer with pH above its isoelectric point. So, it becomes negatively charged, and it will pass through the column. One of the primary advantages for the use of ion chromatography is only one interaction involved during the separation. Another advantage of ion exchange is the predictability of elution patterns. By controlling changes in ionic strength or in pH of the buffer, using different forms of gradient, proteins are eluted differently in a purified concentrated form.
I hope you all like this one about the basic principle behind it. Then comes the affinity chromatography. This method relies on the knowledge of the properties of the proteins. That is, the either antibody or the enzyme will be used in this case. So here you can see there's a glucose binding protein attached to the glucose, and in the affinity chromatography, the use of glucose resins to isolate glucose binding protein conclavin A. So addition of glucose, so glucose binding proteins are released on the addition of glucose. So in this case, basically based on the pH and antibody and antigens that they are playing role. So protein recognized by the antibody and protein not recognized by the antibody. So the red ones are the recognized, the blue ones are the not one. So blue ones will be washed away, and you will have your antibody attached resins in there, and then you can remove the elute with the other pH buffer. So you do it with the other buffer, earlier with the other buffer. So like you have a separated part of it. So there are some drawbacks, that is binding to a ligand antibody can be so strong that very harsh conditions are needed for that. Non-specific binding may also occur, especially where large ligands are required. This non-specific binding can overwhelm the correct binding sites at very high protein concentrations, leading to the pure purifications. Thus, there is often a limit to the protein concentration that can be applied to the affinity column. So, would you like to see the videos of this also? Because it's based on the same principle of pH. So, there is not much difference actually. Would you like to see the picture video for this also, students? Are you awake, everyone? Are you slept because of the boring lecture? No reply. Okay, there's a reply. Okay.
Separation chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
So that was a 3D animation of fusion buffer principle that we have seen in the previous one, that how different pH are being playing role in that. So moreover, it was just a small 3D version of the same. Now comes the HPLC. So this HPLC is an enhanced column method for protein separation. The resin size are more refined in this case. All the chromatographic methods described above can be applied with HPLC column and equipment, but with the HPLC separation of peak is almost always better than the standard one. Um, so in HPLC, there is a large tube containing a small particle beads which act as a stationary phase. In this case, small particles containing with the gel beads, they are acting as a what we say as a gel beads. And on the other hand, the chromatographic bed is composed of the gel beads alone when they are inside the column. The sample is introduced into the injector and then carried into the column by the following solvent. Once the column is there, it's started to separate, adhering or diffusing to the gel. As the solvent is forced into the chromatic bed by the flow rate, the sample separates into various zones of sample components, and these zones are referred to as bands. So chromatographic HPLC, we can see that absorbance at the 220 nanometer, that is 1, 2, 3, 4. So you can check different protein separation at with a raise of in 10 minutes. So let's watch this.
HPLC stands for High Performance Liquid Chromatography, but could equally well stand for High Pressure Liquid Chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase nonpolar. In paper chromatography, the solvent moves along the paper by capillary action. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, a component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in a pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
Outside the reservoir, flow rate of the solvent is set through the computer input and controlled by the pumps. So gel electrophoresis for protein separation, the most popular gel is polyacrylate gel, which forms a three-dimensional molecular sieve. Under denaturing condition, the gel can be dissolved and the size from the 10 to 250 kilodalton, and it is quite powerful. SDS page, the key feature is that SDH is coated with the same electrophoretic mobility, and thus molecular sieving effect is major parameter that causes separation of protein. So gel electrophoresis, we have done it in quite detail with the help of our lecture also, presentation and the virtual lab. So I will not repeat about this, the how your things are separated. Like you can see the large molecules, uh, middle molecules and the small molecules, how they're separated in the porous shell. So this is your STS structures looks like. So your proteins are separated into this different pattern with the large, small, and this one, STS staining, carbon label, STS page. Now this one is important, isoelectric focusing. So this is being separated based on the pH also. So you have one side is high pH of your gel, sorry, of your the paper. So you have molecules which are positive and negative, positive, negative. All molecules are there. So one side is high pH, one side is low pH, and the molecules will decide which side to go. So they will find their isoelectric focusing point. So when we run them on the gel, so they will find where their pH is neutral and your molecules will be separated into that part. And this will further go for the 2D gel to separate them over the gel actually. So this is how your STS pages look like, and this is your isoelectric focused looks like. So it's more dense. You can detect difference of one charge per molecule. So it's quite good one. So let's watch video for this also. 2D gel electrophoresis. I think there is no sound, so I will speak behind that. So you can do it with the help of extract proton. Uh, so you add 1 ml of extra extract pro to cell samples. Then you vortex for 15 minutes. Centrifuge at 12,000 G for 4 degrees Celsius at 4 degrees Celsius for 10 minutes. Then transfer the supernatant to the new tube. Then you take the chloroform and add to your sample, 200 microliter, and shake it for 15 seconds. Incubate at room temperature for 3 minutes. Then again centrifuge for 12,000 G for 4 degrees Celsius for 15 minutes. Then you have your interface. Then you remove the supernatant aqueous phase carefully. Then you add 300 microliter of 100% ethanol. To your sample. Shake it. Then keep it at room temperature for 3 minutes. Again centrifuge at 2000 G for 4 degrees Celsius for 5 minutes. Then divide equally the supernatant into 2 tubes. Now protein will precipitate. So we will add isopropanol to the tubes. We shake it for 15 seconds and incubate at room temperature for 10 minutes. and 2D gel electrophoresis. So in this case, we discard the supernatant carefully. Then we add ice cold 1 ml of ice cold acetone to the precipitate protein. Vortex for 1 minute this palette. So repeat this acetone wash for twice. So let it evaporate. So thank you. That was it to get your protein precipitate from your samples. So in the case of two-dimensional electrophoresis, first we do the isoelectric focusing of a sample like this. Then you put them on the uh, you make it on the horizontal phase with a pH 4 to pH 10. Then you separate them on the dimensional gel, second dimensional gel, and here are the results looks like. So this is your high pH, low pH, and this is a STS page. So this is how you combine them together with the isoelectric focusing and STS page together. I think this is the last video of the same thing, how to run 2D gel. It will be a long one, so watch it with the popcorn at least.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the IPG strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, chaps, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has 2 sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across,
And now you have gel side up. Take your forceps, hold onto the strip on the negative side. You'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, but you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before the step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the Protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Bio-Rad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For Criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. And do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel.
Now that the agarose is solid.
Sorry, students. I have to switch it here off now. I will send you this video. You can watch this video later. My battery is 1% off. I don't have the charger. So this is how you can see your SDS page. And then you can also do multi-top mass spectrometry, which is done to check your proteins at various level. So I will share you all these videos, all these presentations. That's it for today. So I have to leave now because if I don't, uh, because it could stop at any time. So sorry for that. I'm extremely sorry. Wish you all good luck for your future. And what shall I say? I will miss you. You were the best students I had, and I hope you learned a lot from these lectures. I can't think much very quickly actually. So yeah, so just take care, be safe, don't go outside much, and take care of your family and everyone. Um, Stay healthy, keep smiling. Yeah, sorry to act like this at the end because uh, battery is 1%. Okay, thank you very much. I will share you everything by going back at home with the charger, and you can watch it the later part also then. Sorry for that, extremely sorry. Yeah, take care. Bye bye. You will get your certificate soon, and 18th September is the last date for your review. Send it before that, and then we see us soon then. Take care. Bye. बैटरी 1% थी
कोई आपका फ्रेंड है क्या?
नहीं बट हैं
स्टूडेंट्स हैं कौन?
स्टूडेंट्स हैं सारे ऑनलाइन ना यूट्यूब पे आ जाते हैं 20-25 जो हैं तो चैटिंग कर रहे होते
तो
हैं
इनसे पैसे ले रहे हो आप?
मेरे को मिल रहे हैं पैसे एडोफैब्रिका कंपनी है वो बच्चे खुद लेते हैं 1,000 बच्चे एक बच्चे से लेते हैं वो
अच्छा
मेरे को वो देते हैं 10 दिन के 8,000 रुपये देते हैं पढ़ाने के 800 रुपये एक दिन का तो बस यही है अभी तो